So hi everyone, Elliot Rees, Cardiff University. So it's my pleasure today to introduce Sophie Chick. So Sophie is a third-year PhD student. She's supported by a Fieldrose Charitable Trust scholarship. Her work focuses on advancing our understanding of the contribution of rare variants to schizophrenia liability. And she's going to give a talk today on her work on exome sequencing data. So over to you, Sophie.
Yeah. Thanks very much, Elliot, for the introduction. So yeah, I'm Sophie. As Elliot said, yeah, I'm a third year PhD student in the psychosis group at Cardiff. And as part of my Mental Health Research UK funded PhD, I'm studying gene discovery in schizophrenia. To give a little bit of background on what I mean by gene discovery. So schizophrenia is what we call a complex disorder. It sits in between conditions with environmental causes like infections and conditions with genetic causes like rare genetic disorders. And it has some influence from both environment and genetics. So for example, it runs in families, but even identical twins may not have the same outcome depending on their environment. And it's more common in people with certain environmental exposures, but even two people with exactly the same exposure may react completely differently based on their genetics. So complex disorders are both nature and nurture. And in the case of schizophrenia, factors, lifestyle factors, trauma history, substance misuse, and certain infections. And on the genetic side of things, so these are all of the risk factors that have been identified by genetic studies so far. You can see that they cluster into two groups. There are rare large effect risk factors, which each only affect a tiny proportion of people, but have really substantial effects on risk. And there are common small effect risk factors, which are found in much larger proportions of the population, but only individually have very slight effects on risk. And the interesting thing about genetic factors is that by looking at which specific genes are affected, it's possible to get an idea of which biological processes are actually being disrupted in schizophrenia, which in turn could potentially help us inform drug development and treatment of schizophrenia. The most direct way to do this, to identify specific genes, is by looking at rare variants, which are shown in red. Unlike the other types of variants that are plotted here, which basically act over large areas, rare variants are small mutations affecting usually one or a few bases of DNA. And because they only disrupt individual genes, they can be used to implicate single genes that are contributing to risk. And it's this type of variant that I'm working with in my PhD. In rare variant studies, the way that these variants are used to implicate single genes is that so we sequence a population of people with schizophrenia and a population of people without schizophrenia to detect rare variants. And then we compare the rate of rare variants between cases and controls to look for genes where rare variants are associated with having schizophrenia. For example, in gene 1 and gene 2, the rates are similar, as you would expect to see for most genes. But in gene 3, in this example, the rate is much higher in cases. So we would say this gene is significantly associated with schizophrenia and is acting as a genetic risk factor. We can also use trios, which are two parents and an affected child. In this case, we sequence all three members to detect new mutations, and then we compare the rate of new mutations to the rate we'd expect to see in that gene to look for genes where new mutations are associated with schizophrenia. So in gene 3, so if you see 3 new variants in that gene, but you only expect to see, say, 0.2 variants, then again, we'd say that gene is statistically associated with schizophrenia. And this is what I mean by gene discovery. The largest gene discovery study to date, which is important to mention because my work builds off of it, was performed 2 years ago by the Schema Consortium, which is an international consortium. They compared 24,000 schizophrenia cases to 97,000 controls and combined these results with results from 3,400 trios. And they found that 10 genes are significantly associated with schizophrenia, and 22 genes are associated at a slightly lower level of significance. Some really key insights came from this study. I'm just going to highlight one here, which is that of the 10 top genes, 2 genes, GRIN2A and GRIA3, are components of glutamate signaling, which has previously been implicated by different kinds of evidence from pharmacological studies and imaging studies, but this was the first direct genetic confirmation of the role of glutamate signaling in schizophrenia. They also found that, to quote schema, even after excluding significantly associated genes, schizophrenia cases still carry a substantial excess of ultra-rare variants, which indicates that more risk genes await discovery using this approach. In other words, by expanding upon this sample in the future, it will be possible to implicate genes that are currently below this lower threshold and provide more and more insights into schizophrenia biology. So this is what I set out to do in my PhD. I was very fortunate to be given access to a new sample of sequencing data from 4,658 cases and 5,719 controls sequenced at Cardiff University. And in this sample, I firstly evaluated the rates of rare variants in cases and controls. Firstly, I looked in constrained genes, which are a set of genes that are depleted for rare variants in healthy people and that we would expect to be enriched in schizophrenia. This plot basically shows how much higher the rate is in cases than controls, with 1 meaning that there's no difference. And you can see that, as we'd expect, the new sample has a significantly elevated rate of rare variants in cases, which is consistent with what was observed in schema. I also tested for enrichment in previously implicated genes, so all of the genes that are shown, that are named on this plot. And I found that there was really strong enrichment in cases across these genes. So this replicates the enrichment that we saw in schema, and again is in line with what we would expect for a schizophrenia case control sample. So I moved on to combining or meta-analyzing this new sample with schema for the purposes of gene discovery. I did this in 2 stages. Firstly, I combined the 2 case control samples. The plot on the right shows the case control results for benign variants as a validity check. You would expect benign variants not to be associated with schizophrenia, and the plot shows that they follow a null distribution as expected, which means that we can be confident that our analysis is well controlled for factors like different ancestral groups. And I then combined the case control results for each gene with the trio results following schema's approach. So these are the results from the meta-analysis. The novel genes are in bold, so there are 2 novel genes at the higher significance level and 6 at the lower significance level. And I would say that there are 2 key insights from this set of novel genes. The first is related to STAG1. So STAG1 is part of a complex called cohesin, and one of its jobs is to organize DNA in the cell. This is a really fundamental cellular function that's only previously been implicated in rare neurodevelopmental disorders like Cornelia de Lange syndrome. So this indicates that this might be a potential mechanism contributing to schizophrenia as well. And the second is related to SLC6A1, which is just below this higher significance threshold. SLC6A1 is involved in GABA signaling, which similarly to glutamate signaling, which I mentioned earlier, has been implicated in schizophrenia by multiple lines of evidence, but this is the first genetic confirmation of those findings. And this is a component of inhibitory signaling, complementary to glutamate signaling, which is excitatory. So along with the schema results, this highlights the importance of both inhibitory and excitatory signaling in the pathogenesis of schizophrenia. Another implication of these results is their relationship with common genetic risk. So of the 8 novel genes, 2 are also implicated by common variants in schizophrenia. One was previously reported by schema, which is STAG1, and the other is KLC1. So this suggests that rare risk and common risk at least partially converge on the same genes or the same mechanisms, which informs our view of how these different kinds of risk interact with each other. And lastly, I looked at whether any of these novel genes are risk factors for other neuropsychiatric disorders because they're known to overlap genetically. Strikingly, 4 of the novel genes are implicated in other disorders. This is STAG1, SLC6A1, ZIMI-ND11, and SIGREF1. So effectively, half of the novel genes are specific risk factors for schizophrenia, and the other half seem to be acting as more general neurodevelopmental risk factors, conferring risk across disorders. which is informative because it suggests that some of these genes might be impacting mechanisms that are shared between disorders, whereas others are affecting more specific disorder-specific mechanisms. And I think elucidating this cross-disorder overlap more will be an interesting direction for future research. So to summarize, in my PhD, I've analysed a new schizophrenia case control sample, which shows rates of rare variants consistent with previous schizophrenia studies. By combining this sample with the schema sample, I implicated 8 novel genes, highlighting chromatin organization and inhibitory signaling as potential causal mechanisms. These results also highlighted the convergence of rare and common risk, suggesting that they act at least partially through the same genes. And 4 novel genes are implicated across neuropsychiatric disorders, suggesting that many risk factors may not be specific to schizophrenia and informing our view of the overlapping nature of psychiatric genetics. So I'd like to thank Mental Health Research UK and the Phil Jones Charitable Trust for funding my PhD and giving me the opportunity to research this. My supervisors, Elliot and James, my co-authors for this work, the psychosis group, and the participants and their families who contributed to the analysis. And thanks for listening, and I'm happy to take questions.